A tomato plant at the edge of a backyard, the kind that went into the ground in late spring with such promise, is now a shadow of what it was meant to be. The leaves have gone the color of old straw. The stems have lost their rigidity, bending sideways under their own weight. The small clusters of fruit that formed 3 weeks ago have not moved since. They are exactly the size they were when they first appeared, and they will stay that size. Most people see this and feel the particular mix of shame and exhaustion that only gardening produces. They pinch a yellowing leaf between their fingers, shake their head, and begin calculating whether it is even worth trying to save, or whether the better choice is just to pull it, add it to the compost, and tell themselves there was nothing left to be done. But plant physiologists who study cellular stress responses have documented something that changes that calculation entirely. In the overwhelming majority of cases, the failure is not biological. The plant's own machinery is still intact. What is missing is fuel, specific categories of minerals and microbial activators that no plant can manufacture on its own, and that commercial fertilizers deliver in the most expensive form possible. Every one of those nutrients is sitting in the average kitchen right now. Not in a cabinet. In the trash. 5 zero-cost preparations, each made in under 10 minutes from what most households discard before breakfast. That university soil labs have measured. That subsistence farmers across 4 continents have relied on for generations. And that the fertilizer industry has had no commercial incentive to teach. There is no profit margin in a banana peel. There is no patent on fermented rice water, and that, more than anything else, explains why so few home gardeners have ever heard what comes next. To understand why a tomato plant fails in the exact way the one described here has failed, it helps to go back to a German chemist named Justus von Liebig. In 1840, von Liebig published the finding that would eventually reshape the entire agricultural world. He called it the law of the minimum, and the principle was simple enough that it has never been disproven in the century and a half since. A plant does not grow to the level of its most abundant nutrient. It grows to the level of its scarcest one. Von Liebig visualized this as a wooden barrel with staves of unequal height. The barrel holds water only to the level of its shortest stave, regardless of how tall the others are. A tomato plant with abundant nitrogen and potassium but almost no calcium is not a tomato plant with 2 advantages. It is a tomato plant with 1 critical failure. Every other stave in the barrel is irrelevant until the short one is fixed. Every dying tomato plant is a Liebig barrel. There is 1 nutrient, sometimes 2, that has fallen below the threshold the plant needs to continue. And in most residential garden soils, that depleted nutrient is not something exotic. It is potassium, calcium, or nitrogen, drawn down gradually by the same crops growing in the same beds, season after season, never fully restored because the gardener reached for a general-purpose fertilizer formulated for the average garden, not for the specific depletion pattern of this particular soil in this particular yard. The commercial fertilizer industry understood von Liebig's barrel perfectly. The entire synthetic fertilizer business was built on it. Fritz Haber and Karl Bosch cracked the problem of atmospheric nitrogen synthesis in 1913, and within a generation, the chemical approach to soil fertility had become the default, not because it was more effective than what came before it, but because it could be manufactured at scale, branded, priced, and sold through a retail distribution system. What could not be manufactured, packaged, or patented was not suppressed. It simply became commercially invisible. The kitchen pantry is not a retail channel. The banana peel does not have a shelf. What remains in that pantry, however, is a complete soil rescue system. One that addresses every major deficiency category a failing tomato plant is likely to be experiencing. One that begins working within 48 hours. And one that starts with the single most common nutrient failure in residential tomato beds, the failure that most gardeners can identify from 6 feet away if they know the one specific sign to look for. The first method is called the potassium resurrection brew. Before going any further, consider a question that most gardening advice has never bothered to ask. If potassium is the single most important mineral for flowering, fruiting, and root resilience in a tomato plant, why does almost every budget fertilizer on the market deliver it last, in the smallest ratio, and in the least bioavailable form? Tomatoes are potassium-intensive crops. A mature tomato plant in active fruit production draws as much as 400 milligrams of potassium from the soil every single day. That figure comes from research conducted at the University of Florida's Institute of Food and Agricultural Sciences, and it is not a theoretical estimate. It is a measured daily withdrawal from the root zone under normal growing conditions. When that withdrawal is not replenished, the plant makes a decision. It begins to cannibalize the potassium stored in its older leaves, redirecting it toward the developing fruit and the newest growth. The older leaves yellow, then brown, then drop. The stems weaken. The fruit stops sizing. What the gardener reads as general decline is actually the plant executing a very precise triage protocol, robbing its own structure to protect its reproductive future. The sign that distinguishes potassium depletion from other deficiencies is specific. The yellowing starts at the outer edges of the oldest leaves first. The leaf margins go yellow while the center stays green. That pattern, in the lower canopy of a struggling tomato plant, is the Liebig barrel announcing exactly which stave is short. A banana peel contains between 35 and 50 milligrams of potassium per 100 grams of peel material, potassium bound to organic compounds that the plant's root system can process directly, without the conversion steps required for synthetic potassium salts. 3 banana peels submerged in 1 liter of room temperature water and left to soak for 48 hours produce a solution that soil analysis has shown to carry enough bioavailable potassium to begin correcting moderate deficiency within a single watering cycle. Here is how to prepare it. Take 3 banana peels. Fresh or dried, either works, though dried peels release minerals more efficiently. Place them in a jar with 1 liter of water. Cover the jar loosely to allow gas exchange. Leave at room temperature for 48 hours. At the end of that period, remove and compost the peels. Dilute the resulting liquid at a ratio of 1 part brew to 4 parts water. Apply directly to the base of the plant, soaking the root zone thoroughly. Repeat every 7 days. Now here is the common mistake. Most gardeners who hear about banana peel fertilizer boil the peels to speed up the extraction. That is exactly wrong, because heat degrades the potassium-binding organic compounds that make this preparation effective. The potassium becomes partially soluble, but in a form that root membranes cannot efficiently absorb. The cold water soak is slower, but it produces potassium in the organic molecular form that root cell walls evolved to accept. Do not boil. Soak. Within 48 hours of the first application, a tomato plant experiencing active potassium depletion will visibly stop the yellowing progression in its lower canopy. The oldest leaves that have already gone yellow will not recover. Those cells are already gone, but the yellowing front stops advancing. That is the first signal of stabilization, and it is the signal that tells the gardener the plant still has the biological capacity to respond. Potassium stabilization solves one side of the equation for a struggling tomato plant, but there is a second failure that runs underneath the yellowing, invisible in the leaves and invisible in the stems. One that does not show up until the fruit is already forming, and by then it looks like a death sentence. It appears as a soft, dark patch at the base of every developing tomato, spreading inward from the blossom end. It has its own clinical name. And the preparation that corrects it has been sitting in the bowl next to the stove since the last time someone made breakfast. The second method is called the calcium key. Here is something that confuses most gardeners when they first encounter blossom end rot. The plant does not lack calcium because the soil contains no calcium. It lacks calcium because the soil contains calcium that is chemically unavailable. That distinction matters more than almost anything else in vegetable gardening. And it is the distinction that makes a bag of garden lime less useful than what is about to be described. Calcium in soil exists in several chemical forms. The form that plants actually absorb, ionic calcium, Ca2+, in soil solution, requires a specific pH window, adequate moisture for dissolved minerals to move through the soil profile, and the absence of competing ions like magnesium and ammonium that block calcium uptake at the root membrane surface. When any of those conditions fails, the plant develops functional calcium deficiency even in calcium-rich soil. Blossom end rot is the visual signature of that failure, a calcium deficit in the rapidly dividing cells of the developing fruit, causing the cell walls to collapse from the inside. The black rot that appears at the bottom of the tomato is not disease. It is structural failure at the cellular level. Researchers at Cornell University's College of Agriculture and Life Sciences have studied blossom end rot extensively and found that the most reliable correction is not a soil amendment. It is the direct delivery of ionic calcium to the root zone and leaf surface, bypassing the soil chemistry problem that caused the deficiency in the first place. The fastest path to ionic calcium that most households have immediate access to is not a garden center product. It is the eggshell from this morning's breakfast. Eggshells are approximately 94% calcium carbonate. When calcium carbonate is introduced to a mildly acidic solution, one with a pH between 3 and 5, the carbonate reacts with the acid to release ionic calcium directly into the liquid. That ionic calcium can then be applied as a soil drench and absorbed by the root system within hours. The preparation requires one ingredient most gardeners would never think to use: apple cider vinegar. Here is how to prepare it. Collect 10 to 15 eggshells. Rinse them briefly and allow them to dry completely. Crush them as finely as possible. A rolling pin inside a sealed zip bag works well. Place the crushed shells in 1 liter of water. Add 2 tablespoons of raw, unfiltered apple cider vinegar. The mixture will fizz as the acid reacts with the calcium carbonate. That fizzing is not a problem. It is the entire mechanism of the preparation. Stir gently, cover loosely, and allow to sit for 48 hours. Strain the liquid through a cloth or coffee filter to remove undissolved shell particles. Dilute at 1 part solution to 5 parts water. Apply as a soil drench at the base of the plant every 5 to 7 days. Now, here is the common mistake. Most gardeners skip the vinegar entirely. They soak crushed eggshells in plain water, assuming that soaking alone will release the calcium. That is exactly wrong, because calcium carbonate has negligible solubility in neutral water. The soak produces a liquid that looks as though it should work, but contains almost no ionic calcium at all. The fizzing reaction when the vinegar is added is not a sign that something is going wrong. It is the sign that the preparation is working. If there is no fizz, the shells are not releasing their calcium. Do not skip the vinegar. The first signs of correction appear not in the existing damaged fruit. Those are already lost, but in the new blossoms that begin developing after treatment. They set without collapsing. They hold their structure. That is the calcium deficit closing. Blossom health and root strength are now addressed, but a tomato plant that has been under nutritional stress for more than a week will also show a third deficiency, one that announces itself in the newest leaves rather than the oldest ones. When the yellow appears at the growing tip instead of the leaf margins, the plant is running out of the element that makes photosynthesis possible in the first place. And the zero-cost source of it that most gardeners discard every single morning smells nothing like a fertilizer, until it does exactly what a fertilizer does. The third method is called the nitrogen morning tonic. Before explaining how this preparation works, it helps to understand what nitrogen actually does inside a tomato plant. Not in the abstract biochemistry sense, but in the observable physical sense. Nitrogen is the primary building block of chlorophyll. It is the element that makes leaves green rather than yellow. It is the raw material of cell division, which means every new leaf, every extending stem, and every developing root tip requires a continuous and sufficient supply. When nitrogen becomes limiting, the plant cannot manufacture enough chlorophyll to maintain its newest growth, and the first visible symptom appears in the young leaves closest to the growing tip. the leaves that have not yet received the nitrogen redirected from the older tissue below them. The distinction between nitrogen deficiency and potassium deficiency is specific enough to diagnose from observation alone. Potassium deficiency yellows the leaf margins of the oldest leaves first. Nitrogen deficiency yellows the newest leaves uniformly, starting at the tip. Both look like the same kind of dying. They require different treatments entirely. In the 1840s, Justus von Liebig established that nitrogen was the limiting nutrient in most agricultural soils, a finding that ultimately led Fritz Haber and Carl Bosch to develop the synthetic nitrogen synthesis process that transformed the 20th century's food supply. What both Haber and von Liebig understood, and what commercial fertilizer marketing has consistently underemphasized, is that nitrogen is one of the most abundant elements on Earth. The challenge is never availability. The challenge is form. Plants cannot absorb atmospheric nitrogen directly. They require it as ammonium or nitrate, which must either be synthesized chemically or produced by the microbial decomposition of organic nitrogen compounds already present in the soil. Spent coffee grounds contain between 1.5 and 2.1% nitrogen by dry weight. Nitrogen already bound to organic compounds in a form that soil bacteria can convert to plant-available nitrate within 24 to 48 hours of application. 2 tablespoons of spent grounds steeped in 1 liter of water for 24 hours produces a nitrogen-rich soil tea that delivers that conversion directly into the root zone, without the pH disruption that comes from applying dry grounds to the soil surface. Here is how to prepare it. Take 2 level tablespoons of spent coffee grounds, the kind pulled directly from the filter after the morning pot has brewed. Place them in a jar with 1 liter of room temperature water. Cover and leave at room temperature for 24 hours, stirring once at the midpoint of the soak. After 24 hours, strain the liquid through a coffee filter or fine cloth. The resulting liquid will be pale amber to light brown in color. Apply it undiluted directly to the root zone as a soil drench, using the full liter per established tomato plant. Repeat every 10 to 14 days. Now, here is the common mistake. Most gardeners who hear about coffee grounds in the garden immediately try fresh, unbrewed grounds rather than spent ones, reasoning that fresh grounds must contain more. That is exactly wrong, because fresh grounds are concentrated with soluble organic acids that can drop soil pH sharply at the point of application, low enough to cause measurable root tissue damage within the first 24 hours. The brewing process extracts those acids into the liquid that goes into the cup. What remains in spent grounds is the nitrogen content and the mineral fraction, without the chemical aggression. Always use grounds that have already been brewed. The 3 preparations described so far address 3 separate nutrient staves in von Liebig's barrel. For many struggling tomato plants, those 3 alone are enough to halt the visible decline and begin recovery. But for a plant that has been declining in the same bed for more than 1 season, a plant in soil that has hosted tomatoes or other heavy feeders year after year, the mineral corrections will continue to be temporary unless the deeper structural problem is addressed. That problem is not in the soil's mineral content. It is in the soil's population of microscopic organisms that make mineral absorption possible in the first place. And it is a problem that no chemical fertilizer was ever designed to solve. The 4th method is called the living water. What most gardeners are never taught, and what changes the entire framework for understanding why the same beds produce diminishing yields season after season, is that a tomato plant does not feed directly from soil minerals the way a sponge absorbs water. It feeds through relationships. The root system of a healthy tomato plant is surrounded by and intertwined with billions of soil microorganisms, primarily bacteria and mycorrhizal fungi, that act as translators between the raw mineral content of the soil and the chemical forms the plant's roots can absorb. Without those organisms, minerals in the soil remain locked in forms the plant cannot use. Remove that microbial community through repeated synthetic fertilizer applications, through excessive soil disturbance, or simply through years of chemical inputs that suppress bacterial populations in the root zone, and the plant becomes functionally deficient regardless of what the soil test says is present. In the 1980s, Dr. Teruo Higa, a researcher at the University of the Ryukyus in Okinawa, Japan, began systematically documenting populations of what he called effective microorganisms. Specific strains of lactic acid bacteria, yeasts, and photosynthetic bacteria that colonize soil and dramatically accelerate the rate at which organic compounds are converted to plant-available mineral forms. His research, which was eventually incorporated into agricultural programs across more than 150 countries, demonstrated something that ran against decades of commercial fertilizer science. The fastest way to restore mineral uptake in depleted soil was not to apply more minerals. It was to restore the organisms responsible for making minerals available. Fermented rice water is a traditional preparation that produces exactly such a culture, and it does so from materials available in virtually every kitchen in the world. The starch coating on the surface of unwashed rice grains hosts native populations of lactobacillus bacteria. When those grains are rinsed in cool water and that rinse water is left to sit at room temperature, the bacteria begin fermenting the dissolved starches. After 3 to 5 days, the liquid contains a measurable and active population of beneficial bacteria in the same functional categories that Higa's research identified. Korean natural farming practitioners have used this preparation for centuries, documenting its effects on soil health across generations of cultivation long before the Western scientific community began measuring the same outcomes. Here is how to prepare it. Rinse 1 cup of uncooked, unwashed white rice, the kind that has not been pre-rinsed or processed, in 2 cups of room temperature water for 1 to 2 minutes, stirring to release the surface starch. Reserve the rinse water. Do not rinse the rice under hot water at any stage, as heat will kill the bacterial cultures before fermentation can begin. Leave the cloudy rinse water in a loosely covered jar at room temperature for 3 to 5 days. During that period, the liquid will develop a faintly sour smell. This is the signature of active lactobacillus fermentation, the same organism responsible for yogurt and sourdough bread. At the end of the fermentation window, dilute at 1 part rice water to 10 parts fresh water. Apply as a soil drench, working the liquid gently into the top 4 to 5 inches of soil around the plant base. Repeat every 7 days. Now, here is the common mistake. Most gardeners who hear about fermented rice water allow it to ferment for a week, sometimes 2, on the assumption that longer fermentation produces a more potent preparation. That is exactly wrong, because beyond 5 days, the lactic acid concentration in the liquid rises to levels that function as a biological soil sterilant rather than a microbial inoculant. At that concentration, the preparation does not add beneficial organisms to the soil. It kills the ones already present. The 3 to 5 day window produces maximum bacterial density and minimum acid concentration. Beyond that window, the preparation reverses its own purpose. Set a reminder. Stop it on time. The living water restores the invisible workforce that makes all mineral delivery possible. But there is a 5th category of depletion that none of the preceding methods addresses on its own. Not a macronutrient, not a microbial problem, but a slow cascade through the trace elements that function as cofactors in nearly every enzymatic reaction in the plant. It shows as general, gradual decline with no single dominant symptom. The plant that simply seems to be running out of energy despite adequate water and visible nutrition. And the preparation that addresses it most efficiently has been collected and discarded in the same kitchen as everything else in this video, before nearly every home-cooked meal for years. The 5th method is called the golden sulfur tonic. The question worth asking before this method is explained is one that almost no mainstream gardening publication has raised directly. If trace mineral deficiency is one of the most consistent causes of declining tomato yield in established home garden beds, and if those trace minerals are present in the organic matter flowing through the average kitchen every single day, why has the default recommendation always been to purchase a commercial micronutrient supplement rather than recover what is already being discarded? Onion skins. the dry, papery outer layers peeled away and thrown into the trash before virtually every home-cooked meal, contain a trace mineral profile that plant scientists at the University of Agricultural Sciences in Bangalore, India, have documented in published research. The dry outer layers of a single medium-sized onion carry measurable concentrations of quercetin, a flavonoid compound that in soil solution functions as a chelating agent, a molecule that binds ionic trace minerals including iron, zinc, and manganese, and holds them in forms that root cell membranes can transport directly rather than converting. Beyond the quercetin, onion skins carry calcium, phosphorus, and sulfur compounds that together form a broad-spectrum trace element supplement. Sulfur, specifically, is the mineral most consistently overlooked in home garden fertility programs. In a tomato plant, sulfur is required for the synthesis of two essential amino acids, cysteine and methionine, without which protein formation and cellular enzyme function both degrade progressively. Sulfur deficiency presents as pale, uniform yellowing across young leaves, often confused with nitrogen deficiency, but occurring even when nitrogen applications have been recent. The distinction is in the uniformity. Sulfur deficiency yellows the entire young leaf, including the veins. Nitrogen deficiency leaves the veins green longer. The plant looks similar. The treatments are entirely different. Here is how to prepare it. Take a double handful of dry onion skins, the outer, papery layers only, not the fleshy interior layers of the onion. Place them in a pot with 1 liter of water. Bring the water to a boil, then immediately reduce the heat and allow to simmer for 10 minutes. Remove from heat and allow the liquid to cool fully to room temperature. The resulting liquid will be a deep golden amber color. Strain out all solid material. Dilute the concentrate at 1 part tonic to 5 parts water for a soil drench application, applied to the root zone of the plant and worked gently into the top few inches of soil. For plants showing advanced trace mineral depletion, mottled coloring across young leaves, distorted growing tips, reduced blossom set, dilute at 1 part to 8 parts, and apply as a foliar spray in the early morning, allowing the leaves to dry before midday sun arrives. Repeat every 7 to 10 days. Now here is the common mistake. Most gardeners collect onion skins over several weeks, storing them in a closed plastic bag or container until they have enough to brew. By the time they use them, some of those peels have developed surface mold that is not always visible to the naked eye. That is exactly wrong, because even partially moldy dry skins will release fungal spores and mycotoxins into the brew that can introduce soil pathogens far more damaging than the deficiency the preparation was meant to correct. Onion skins for this preparation must be fully dry and visually clean. Collect them in a paper bag or open basket, never sealed plastic, to allow air circulation. Discard any skin that shows discoloration beyond the natural amber and gold of the dry outer layer. If there is any doubt about a peel, discard it. The preparation is only as clean as its least clean ingredient. What these 5 preparations represent, taken together, is not a collection of home remedies assembled from old gardening folklore. It is a complete fertility cycle. Potassium for vascular function and fruit set, calcium for cellular integrity and blossom health, nitrogen for photosynthetic capacity and new growth, living microorganisms for mineral conversion and root health, and trace elements for every enzymatic process that connects those nutrients into growth. A commercial fertilizer product at $40 a bag delivers a fraction of this spectrum in forms engineered for shelf stability and transportation rather than root uptake. 5 kitchen preparations made in under an hour deliver the complete cycle in the bioavailable forms that tomato roots evolved over 10,000 years of cultivation to accept. There is a principle underneath these 5 methods that is worth naming directly, because it is the principle the fertilizer industry would prefer most home gardeners never arrive at on their own. Every one of these preparations works because it is returning something to the soil that the kitchen itself extracted from the soil in the first place. The potassium in a banana peel came from mineral-rich tropical soil. The calcium in an eggshell was drawn from the same calcium cycle that built the shell from the inside out. The nitrogen in spent coffee grounds was fixed from the atmosphere by soil bacteria generations back. These are not new inputs introduced into a system from outside. They are recovered outputs, materials that belong in the soil, that have always belonged in the soil, and that the modern habit of landfill and garbage disposal has been quietly redirecting away from it for decades. The fertilizer industry did not create plant nutrition. It packaged it, priced it, and taught several generations of gardeners to outsource something their grandparents understood how to do themselves. The banana peel that was composted or discarded every morning for 30 years was not waste. It was a potassium supplement waiting to be returned to the ground it came from. The eggshells, the coffee grounds, the rice water, the onion skins, all of it flowing toward the landfill every week, while the same gardeners who produced it drove to the garden center to buy back a chemical approximation of what they had already thrown away. The tomato plant at the edge of the backyard is not a failure. It is a Liebig barrel with a few short staves. Lengthen the staves, one preparation at a time, and the barrel fills. If any of these rang true, leave a note below about which one you will try first. Share this with a fellow gardener who has been losing more plants than they have been saving. And keep the old knowledge alive, because every kitchen that turns its scraps back into the soil is one more small piece of something worth holding on to.